Hi, my name is Emmy Bruns. I'd like to tell you about a recent paper, Is There a Disease-Free Halo at Species Range Limits? The Codistribution of anther-smut disease and its host species. Ecology is the study of abundance and distribution. As a disease ecologist, I'm interested in how infectious disease affects the abundance and distribution of hosts. When we know that disease can reduce host abundance, the effect of disease on host distribution is actually a trickier problem. A classic view of species range distributions is the central abundance view. Density declines towards the range edge where conditions are generally crummier. If disease can drive these marginal populations to extinction, however, it could restrict host populations. However, disease must be able to persist in these low-density populations for this to occur. An important concept in disease ecology is threshold density, the minimum host density required to sustain an epidemic. This is a common feature of diseases with density-dependent or mass action transmission. Diseases with threshold densities will not be able to persist in low-density populations near the range edge, resulting in a disease-free halo at the distributional limits of a host species. However, not all diseases have threshold densities. Diseases that are spread by vectors or through sexual transmission tend to be independent of density because contact rates are not density dependent. Theory predicts that diseases with this type of frequency-dependent transmission can actually persist in low-density populations at range margins. However, we currently lack empirical studies of the distribution of frequency-dependent diseases at range limits. My colleagues and I decided to test this prediction by studying the co-distribution of anther-smut disease on alpine plants. Anther-smut disease is caused by fungi in the genus Microbotrium. The fungus hijacks the plant's reproductive system, causing it to produce spores in place of pollen. Infected plants are rendered completely sterile and are unable to produce either pollen or seed. It is an ideal system for studying disease distribution in nature because of its easy visual detection. Moreover, since the disease sterilizes but does not kill its host, we can estimate prevalence from simple field counts. Most importantly, transmission is facilitated by insect pollinators visiting diseased plants. This makes it likely that transmission is frequency dependent, and we therefore predict that there would be no disease-free halo at the range margins in this system. Alpine plants have distinct elevational ranges that can be easily traversed in a single day. It is also possible to replicate transects in several different montane valleys. Here we took a replicate transect approach and mapped the distribution of plants and their associated anther-smut diseases across 59 valley transects in the Maritime Alps region of Italy. This area has steep elevational gradients, as well as a high diversity of both host and pathogen species. We mapped the co-distribution of anther-smut disease on 4 different species: Silene latifolia, Silene vulgaris, Dianthus ficardis, and Dianthus pavonius. Each host species in this group is infected by its own specific anther-smut pathogen. Within each transect, we recorded GPS points every 100 meters in elevation. We recorded the location of the first population we encountered and the first disease population encountered at each of these 100-meter increments. Within a site, we also counted all plants within a 100 square meter area to get host density and disease prevalence. This allowed us to estimate host occurrence and density, as well as disease incidence and prevalence. These black dots represent all of our sampling locations. In total, we mapped over 700 host populations. We found distinct elevational range limits for each host species. These graphs show host occurrence, the proportion of transects where host populations were found. The 2 Silene species were abundant at lower elevations and had clear upper range limits, shown with the blue arrows. The 2 Dianthus species were abundant at high elevations and had clear lower range limits. We also found that host density, the number of plants within a population, declined towards the elevational range limits, as predicted by the central abundance hypothesis. Disease incidence, measured as the proportion of populations with disease, was significantly greater than 0 at the elevational range limits of both Silene latifolia and Dianthus pavonius. Moreover, disease prevalence within populations did not decline at the range margin. Instead, in all 4 species, disease prevalence tended to increase near the range limit. We also examined the relationship between host density and disease incidence to see if there was evidence for threshold density for persistence. We found no evidence of a density threshold for disease in either Silene latifolia or Dianthus pavonius. In these species, incidence was predicted to be greater than 0 in populations with as few as 5 individuals. This is shown by the 95% confidence intervals that are not overlapping 0. In the other two species, population incidence was greater than 0 in populations with 10 or more individuals. Overall, we found no evidence to support a disease-free halo at host elevational range limits. Instead, we found that disease was present at the range limits in all 4 species. In addition, disease prevalence within populations was actually higher at the range limit than in the range center. These results are important because it means diseases with frequency-dependent transmission could have an effect on host distributions.